For decades, there have been an enormous variety of pollutants contaminating our freshwater lakes, rivers, streams, and wetlands. Pharmaceuticals are only a fraction of the pollutants threatening our aquatic ecosystems. They come from a broad range of organic and inorganic compounds. From this broad range of pharmaceuticals, specific functions and their effects on the biological systems have categorized certain compounds to be known as endocrine disruptors. To better understand concepts regarding endocrine disruptors in relation to limnology, several factors must be examined. Potential sources of the pollutants must be determined. The function of these compounds and their effects on biological systems, as well as their movement through various trophic levels through biomagnification, may cause significant effects on the aquatic ecosystem as a whole. Concepts of mitigation and remediation may play a fundamental role in the survival of our many threatened ecosystems. Okay, so before you can understand anything about endocrine disruptors and the problems they pose in the environment, you have to understand exactly what the endocrine system is. The endocrine system is one of your body's main biological functions. It involves glands that produce and secrete hormones, which in turn influence almost every cell and organ in the body. This system regulates mood, growth and development, metabolism, as well as sexual function and reproduction. Endocrine disruptors are chemical compounds that get introduced to organisms or cells from an outside source. Once they're inside a biological system, they can cause many changes. They mimic or act as natural hormones do. They can also neutralize or repel natural hormones while affecting their degradation and production. All these chemical changes can result in many negative effects in humans and animals, such as cancerous tumors, brain problems, deformation of body parts, as well as affecting sexual development. So once you're familiar with the endocrine system and what endocrine disrupting chemicals are capable of, it becomes apparent that the release of these types of chemicals in the environment can become a very serious issue. Chemicals possessing endocrine disrupting capabilities can be found everywhere. In our homes, in our food, in our cars, and at work. They were first recognized by science back in the 1950s with the use of a pesticide known as how do you say that?
Its name is dichloro-diphenyl trichloroethane, for short, DDT.
DDT. DDT was used as a pesticide in World War II to protect troops and civilians against malaria spread by mosquitoes. Bisphenol A, or BPA, is another example of an endocrine disrupting chemical that can be found everywhere. It's an industrial chemical used to make polycarbonate plastic for water bottles and food containers, as well as protective lining inside metal cans. The chemical can leach into food from tin can linings, from plastic food covers and water bottles, especially when exposed to heat. Estrogen from birth control contraceptives also have endocrine disrupting capabilities.
Endocrine disruptors are often deposited into our water systems from wastewater effluent. Most endocrine disruptors aren't completely metabolized by the human body and are often passed through waste, making their way to local wastewater treatment facilities. Wastewater facilities are able to remove some of these compounds, yet there are still trace amounts present in effluent, which is then released to surface waters. From there, these chemicals are distributed throughout the aquatic ecosystem by dissolving in water and absorbing to sediments and particulates.
In general, endocrine disruptors have a low water solubility. However, trace amounts can still be found dissolved in the water. Sediments, on the other hand, tend to play a larger role in the movement and storage of these substances. Small particulates, primarily clay, have a larger surface area in which endocrine disruptors can attach and absorb to. For this reason, lake bottoms with a large composition of clays will act as a sink, storing these chemicals for years. Not only does the small size of the clay allow for longer storage, but it also helps the movement of these substances from one place to another. Due to the light weight of the clays, they can be easily swept up and moved around during times of turbulent flow caused by heavy rainfall or spring melt. These turbulent flows can carry contaminated sediments downstream, depositing them around the mouths and centers of lakes.
A study in Japan tested harbors impacted by intense human and industrial activity to determine the distribution and concentration of endocrine disruptors in harbor sediments. This study showed that high levels of endocrine disruptors were present in harbors being used as ports. Results show that concentrations tend to be higher in the middle of the harbors, most likely due to sediments settling out of the water and acting as a sink. By analyzing the core samples taken from the harbor sediments, it is possible to relate concentrations of endocrine disruptors to common industrial practices of the time. Results showed very little traces before 1950, but increased dramatically from 1960 to 1990, most likely due to the utilization of agricultural chemicals such as PCP and CNP. This study just proves that these chemicals are deposited into sediment and have the capability of being stored for long periods of time, allowing aquatic organisms to be exposed to these chemicals throughout their entire life. Another study was conducted in Shiwa Lake, Korea, to determine if there was a correlation between season and spatial distribution of nonylphenol. It was observed that concentrations in water tend to fluctuate with a change in season, while there was no observed trend between season and sediment concentrations. High concentrations were observed in summer months, which may be a result of higher degradation rates of AEPs under hotter temperatures. It was noted that concentrations decreased farther away from industrial sites, showing that industry was the main source of contamination in this particular lake. A correlation between nonylphenol concentrations and phosphorus content was also observed. Distribution level of endocrine disruptors depend on a number of factors, such as the activity of local microbiology, sediment characteristics, sediment drift, sedimentation rate, and dissolved oxygen content.
Animals, including humans, can and do accumulate some of these chemicals in their fat, and often pass them along to offspring and predators as part of the food chain. The increasing concentration of chemicals accumulating through each level of the food chain is known as biomagnification. A completed survey of the river Aroni in central Italy is a great example of biomagnification. Concentrations in the river water were found to be low, at the nanogram per liter level. Depending on their distance from the contamination source, level of contaminants found in sediment ranged from 10 to 200 micrograms per kilogram. Concentrations in macroinvertebrates such as crayfish, mussels, and snails were found to range from 60 to 400 micrograms per kilogram, and the highest concentration of chemical pollutants were found near the top of the food chain in the fish. In England, male fish living close to municipal sewage outlets were found to have both male and female sex characteristics.
This fish has characteristics of both males and females.
Their livers produced vitalogenin, a female egg yolk protein not normally found in males. The fish living close to the sewage outlet had severe abnormalities, while the fish living farther downstream had less severe symptoms. Several different chemicals and the breakdown products of them found in detergents and plastics are suspected of causing these feminizing effects. Dredging is an operation carried out by using heavy equipment with the purpose of gathering up bottom sediments from shallow seas or freshwater areas and disposing of them at a different location. This process can be used to remove contaminated sediment from lake and riverbeds. The removal of PCB-contaminated sediments in an area of Lake Huron during the year 2000 and 2001 proved to be a success in the reduction of PCB concentrations in the local fish population. During April 1996, 11 male walleye and 10 female walleye were caught in Saginaw Bay, Lake Huron. The individual PCB determinations were made for each fish. Concentration results for the males averaged at 7.95 milligrams per kilogram and 7.13 milligrams per kilogram for the females. In a remediation effort during 2000 and 2001, contaminated sediments were removed from the Saginaw River through a dredging process. In April 2007, 10 male and 10 female walleye were caught from the same area and individual PCB determinations were made for each fish once again. The second time around, the PCB concentration levels averaged at 1.58 milligram per kilogram for the males and 0.55 milligram per kilogram for the females. The dredging remediation effort appears to have been effective in lowering the PCB concentrations found in walleye by 80% from 1996 to 1997.
Seeing as most of the endocrine disruptors in our environment are a product of human processes, such as the use of medicines and industrial production, it is most likely that these chemicals will make their way into municipal wastewater systems and eventually to a wastewater treatment facility. Activated sludge treatment processes carried out in these facilities convert organics into biomass, which can then be removed. Unfortunately, this common method of water treatment will only remove around 85% of these hormone-mimicking substances. This results in contamination of surface waters. The use of UV light to treat wastewater is very effective for the removal of endocrine-disrupting chemicals. A study was carried out to determine if BPA would degrade with the use of UV technology. With medium-pressure mercury UV lamps, a 5 to 25% reduction of BPA was observed. The same samples were exposed to UV and hydrogen peroxide resulted in a greater than 90% decline in BPA and a nearly 100% reduction in estrogenic activity. This type of treatment can be found in some facilities but is not implemented due to high expense.